Hello everyone, in this video I'll be explaining on the application, advantages, limitation and enhancement of bright field microscope. Coming first to the bright field microscope, it is used to visualize and study the animal cells and also the plant cells, which can be used to visualize and study under the bright field microscope. It can also be used to visualize and study the morphologies of bacteria. Example in the gram staining and AFB staining, where in the gram staining it is used to differentiate the gram positive and gram negative bacteria, where it is differentiated based on the color, and also the shape and arrangement of the bacteria can also be viewed under the bright field microscope. Next, it can also be used to identify parasitic protozoans such as paramecium or the malarial parasite, which can be viewed in the blood sample of the patient suffering from malaria and can be observed under the bright field microscope. Coming to the advantages of bright field microscope, it includes simplicity of setup with only basic equipment required. Living cells can be seen with bright field microscopes. It is simple to use with few adjustments involved while viewing the image, so it doesn't need any additional or complicated adjustment for operating the bright field microscope. It can be used to view both stained and unstained particles. That means it can not only view the stained particles under the microscope, but it can also view those particles which are not stained. The optics of the microscope do not alter the color of the specimen. The microscope can be adjusted and modified for better viewing, such as installing a camera to form a digital microscope, or in the way image illumination is done, such as by use of fluorochromes on the specimen and viewing under a dark environment, forming a dark field microscope. Next, coming to the limitations of bright field microscope, it includes only fixed specimens can be viewed under the bright field microscope. It has low contrast, hence most specimens must be stained to be visualized. Use of oil immersion may distort the image. The use of cover slip may damage the specimen, while in some experiments where cover slip is placed upon the specimen. The microscope needs a strong light source for magnification or viewing the particles under the microscope. Samples that are naturally colorless and transparent cannot be seen well. Next to the enhancement of bright field microscope, it includes reducing or increasing the amount of light source by the iris diaphragm. Use of an oil immersion objective lens and special immersion oil placed on a glass cover over the specimen. Immersion oil has the same refraction as glass and improves the resolution of the observed specimen. Use of sample staining method for use in microbiology, such as simple stains, methylene blue, safranin, or crystal violet, and differential staining such as negative stain, flagellar stain, and endospore stain. Use of a colored, usually blue, or polarizing filter on the light source to highlight the features not visible under white light. The use of filter is especially useful with mineral samples. So that is the application, limitation, advantages and enhancement of bright field microscope, an optical microscope which is most commonly used in the lab.